This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 925, recorded on August 5th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. I have no idea what the weather's like, except that it's puffy clouds and blue sky. So, it says it's 85 degrees Fahrenheit, and I'll have to figure that out in Celsius from the Norwegian weather app, telling me that it's 30, feels like 33 Celsius. There you go.
It's 31°C here and quite humid.
Oh, yeah, it's humid here, too.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We have 99 degrees again today, headed for 100. We wouldn't want to break that record. We have a hot summer going on. It's a little hazy. I'm okay. It's good.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and the temp is too damn high. It's
TDH?
Yes, right. So it's nominally it's 91 Fahrenheit, 33 C. The heat index, so the dew point's 70 Fahrenheit. The heat index is 97, which is 36 Celsius. So it's hot and sticky here in Massachusetts.
Have you been doing any flying lately, Alan?
A little bit, yeah. No major trips, but it's an interesting time of year to fly because the air is less dense because it's hot.
So how
does that impact? What did you say the dew point was?
Dew point is 70 here, Fahrenheit.
So when the air is like that, does that impact? Do you have to go longer to take off, or what does that do?
Yeah, so it raises the density altitude, so the altitude the wing thinks it's at. But on balance, I prefer it over flying in the winter around here because I don't have to go out onto the ramp when it's 5 degrees Fahrenheit and blowing wind and yeah.
So what is the speed where you pull up? V1 is that?
That's VR. VR, you'd like that. That's the rotation speed.
And does that change with the temperature? That's what I wanted to ask.
It can. For the planes that I fly, the difference is not significant enough that you'd worry about it.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is also very hot, sunny, and humid here, 88 Fahrenheit, 31 Celsius. Not sad about being away from that right now.
And by the way, congratulations on your Jeopardy victory, Brianne.
Yes.
Mm-hmm.
Very cool.
Thank you. It was so much fun.
Sure.
In fact, I did have someone tell me on Twitter that they found it very strange to see me on Jeopardy and have me not tell them the weather.
That's great.
So they actually asked you which Greek letter was skipped in the naming of the variants.
They did.
I wonder if that was just because you were there.
I don't think so. They told us in some of the legal things at the beginning, all of the things they do to randomize questions. So based on that, I would say not.
But you were ready, man, right?
I was very ready.
That's cool. Very good.
And for those who haven't seen it yet, Brianne, which Greek letter was skipped?
It was Xi.
Xi.
In fact, both new and Xi. But
we went through that once because it had some bad associations, potentially politically incorrect associations.
Correct.
So this week I had 3 visitors here at the incubator. 2 people I met at meetings, one person from ASV, one person from the BAT meeting, and then a local physician and her husband took me out to lunch yesterday, and they brought me a T-shirt. And this is just awesome. Look at this.
Already? Nice. That is great.
Nice.
Can you imagine? She said, you can tell I listen, can't you?
That's great.
The Science Will Break Your Heart, the ASV title. Oh, my gosh. That's great. Thank you. That's Dr. Jackie Mayo, and she is a local internist. And I'm going to get her on to talk about her experiences. It's very interesting to hear how she's been dealing with things. So thank you for coming by. And also she made a nice contribution to Microbe TV. So thank you very much for that. And if anyone wants to visit, we're open. Someone came in today. I forgot her name. I'm sorry. But she said, "Are you the only one here?" I said, "Yeah, I'm the only one here." But we have connections to the world. We can reach many people. All right, we have a couple of updates since we haven't been together for a couple of weeks. I thought many things have been happening. And one of them, of course, well, viruses are in the news. You should listen to this podcast, This Week in Virology.
I'll tell you what, this episode's got everything.
It's got everything.
Yes, it does.
So monkeypox, of course, is accelerating. And I have to say, Rich, I hear you in my head, and we did the same thing. Oh, this is going to burn out. We were wrong. It's not going to burn out.
I know. I've been, God.
We based it on what happened before, right? The other little outbreaks.
But, we complain about others never learning, okay?
Yeah, it's true.
But I'm surprised.
Oh, viruses always surprise.
The fact that I'm surprised, I think, shows how little I know about public health. Okay, I know something about how the virus works and that kind of stuff, but people is another realm. Okay, and I don't really, I don't fully understand. I don't understand people at all. Okay, in particular. On a global level or in a public health context. And this is all about public health.
It is.
And just so you're not out there on a limb alone with your bad prediction, I wasn't on that episode, but at the time I was thinking that, monkeypox, I mean, that's not going to go very far. So I'm totally with you on this is going to burn out. And totally wrong.
And I think that one of the sentences I've said the most frequently over the past couple of years is, well, people I can't predict.
Yes.
So I hear you on that too.
Yes.
So I have a couple of links that I thought were useful because people have a lot of questions. And they're both at the CDC. One of them is how it spreads, which I think is very good.
Yes.
Because it can spread through anyone through close personal skin-to-skin contact. Contact with rash, scabs, body fluids, touching objects that have been used. Many people are asking, how about a blanket in a hotel? Yeah, they don't wash the blanket, the thing on top, the bedspread. That could be intimate contact. And I will let you read their other word for anus, which I think is a riot being on a CDC website, right?
Yeah. Same.
Well,
unbelievable. Wow. I think they're trying to connect with people, right?
Absolutely.
Their communication department has to use the language that is going to reach the audience.
Right.
So, yeah.
But, CDC is a government organization. I could see some government people getting upset, right, about this, but I'm glad it's there. I don't have a problem. Hugging, massage, kissing, prolonged face-to-face contact. A pregnant person can spread virus to their fetus through the placenta. Infected animals by being scratched or bitten. If a person with monkeypox can spread it to others from the time symptoms start until the rash has fully healed, this is typically 2 to 4 weeks. But they say scientists are still researching if the virus can spread if someone has no symptoms, how often it's spread through respiratory secretions, or when a person with symptoms might be more likely to spread through respiratory secretions, whether it can be spread through semen, vaginal fluids, urine, or feces. So a lot of questions.
I really liked that they did that at the end. I don't remember seeing things like that before with the little section of scientists are still researching.
Yeah.
I think that's really helpful to the public and also kind of really helpful to remind people that we are still researching all of these things. We don't
know everything.
Right, exactly.
Right. There was
can we say, I'm sorry, go ahead, Kathy.
I was going to say, there was a news story this morning that basically picked up on all these things, but also went a little deeper into the skin-to-skin contact, and that it really has to be pretty rough contact. And that fits with what Daniel's told us on the clinical updates for the clinicians. If you were going to, swab a lesion, you have to be pretty vigorous. And so that was interesting to hear because, I wonder about this blankets and fabrics thing, but I think if there was pretty rough contact with a blanket or fabric, then maybe some virus could come out of the lesion that way, but just touching it, maybe not so much. I don't know. Go ahead, Rich.
Rich,
in
how
the
long?
absence of, go ahead.
Go ahead. I just want to know how long would infectivity remain on a surface with this virus?
Well, vaccinia virus you can lyophilize.
Right.
Okay, so this is a tough nut.
Right.
Okay, it's not as susceptible to the environment as a lot of other viruses.
But it's not as contagious as smallpox.
Apparently not. Well, smallpox was transmitted primarily as a respiratory infection,
Right.
is my understanding.
Right.
Okay,
But
And
there
this
was
seems-
that rather famous blanket incident.
Oh
Yes.
yeah. I mean, it's well documented that any of these viruses can be transmitted on fomites, and they're I can't give you a number on how stable, but they relative to other viruses, they are environmentally quite stable. Okay, yeah, I think I got to learn to hedge my bets better, but
Yeah.
Current state of knowledge, this is, I would say, overall a lot less transmissible than something like SARS that we've been dealing with, requiring much more intimate contact than SARS. And current data does not, let's put it this way, there's not an abundance of evidence, or any for that matter, that I know of, for asymptomatic transmission. Okay? And that's huge.
Yeah.
Okay? Because that means that if you know you're sick, you can take yourself out of circulation. That's
Yes.
really, I mean, that was important with SARS-1, right?
Yes.
Okay. Now, I don't know what they mean by symptomatic, because there's a prodrome where you got a fever and nausea and swollen lymph nodes before the rash shows up. And that can be a few days. I don't know if they're counting that as symptoms or not. And I also don't know whether you are contagious during the prodrome. Okay. It sounds to me like most of this transmission is from lesions, because there's virus in the lesions. And I assume that one of the reasons that it has to be as rough as it is that typically, at least from what I know about pox viruses, you have to do more than just drop it on the skin. It has to get into a fresh abrasion. Okay, so that's where the roughness comes in. Probably easier with a mucous membrane, okay, than with skin. But we're talking about probably. Now I'm making this up, but my guess is, based on what I know, that a majority, a very significant amount of the transmission is virus from active lesions getting into open skin or mucous membrane on a recipient, requiring very close contact.
Right. The thing I heard did say a pretty rough abrasion, and then into an open or new abrasion in the recipient skin.
There are now cases in all kinds of people, right? So not just gay people, but it's heterosexual. Even kids apparently are getting infected. So
as far as I know, the virus doesn't care how old you are or what your sex is.
No.
Or even how it was transmitted.
Yeah.
Right.
Nope.
So this is, I can certainly picture kids on a playground, wrestling or playing tag or something, and abrasions. I mean, if you have been around kids, you know that they're pretty much always have some kind of abrasions somewhere, because they're,
So someone asked us this week, but what about wrestlers? Yeah, for sure, right?
Yeah, absolutely.
Sure.
I mean, they get herpes gladiatorum, right?
Yeah.
Skin infection. So, Rich, I wanted to ask you, molluscum contagiosum, would that confer any cross protection, do you think?
I don't think so. It's in a different species. I don't know what the taxons are now, but the orthopox viruses that include smallpox, monkeypox, vaccinia, ectromelia, which is mousepox, and cowpox, I think that's all of them, I'm not sure, are all serologically cross-reactive. Okay? And that's important in this case, because smallpox, we have a bunch of vaccines for smallpox, and it's cross-reactive with monkeypox. Okay? So smallpox, monkeypox, vaccinia, which is the vaccine, that's relevant. molluscum is quite a different, same family of viruses, but quite a different beast. So as far as I know, there is no cross-reactivity.
The other CDC page I like is a page called Considerations for Monkeypox Vaccination. So if you have questions about vaccines, you can find out, there are 2 vaccines, Genomes and ACAM 2000. We've talked about those before. Now it's interesting. They say, Rich, Genomes has been approved by FDA for the prevention of monkeypox infection. So that must have been a long time ago, right?
Must have been.
Because it didn't happen in this recent outbreak, right?
Not that I'm aware of. This is the kind of stuff we can ask
Michael.
about.
So we're going to have someone next week on TWiV who knows all about these 2 vaccines.
Yeah.
And ACAM 2000 is a smallpox vaccine.
It is a smallpox vaccine. And we can ask Mike about all this stuff, but I have very little question, but that both the drugs and the vaccines will be essentially equally effective against monkeypox and smallpox. But when you get into regulation and et cetera, they have questions and concerns that is fine with me because they're careful. Think thalidomide, okay? Yeah. So that's, it's important. But I would have a hard time. I can't believe that all of these therapies and vaccines aren't essentially equally effective against smallpox and monkeypox.
So there's a lot of information on this page, and I think it's interesting that in some cases you can use the vaccines post-exposure. Right? I think they say within 4 days of exposure you could get vaccinated and still have, and it's kind of like the rabies vaccine, right? You can get it post-bite.
Yeah, and I think that they know that from the experience with smallpox.
Yeah.
Right.
Right. All right, so we'll put a link to that. Yeah, and then
there's the case trends.
Yeah, the CDC has
here,
case trends. Yeah,
how wrong Rich and Vincent were.
All of us. Yeah.
Well,
us,
except
yes.
that so I looked this up because this is what I really want to see is the trends. I want to see an epi curve, and well, I don't know. Man, it's too early. I was trying to imagine that maybe it was coming down relative to a prior week, but. Maybe that week is artificially high. I don't know.
I found it to be a rather impressive epi curve.
Yes.
What I think is interesting.
Notice they take the weekends off.
Yeah.
Yeah. The numbers are really interesting. Total confirmed cases globally, 26,864. Total cases in countries that have not historically reported monkeypox, 26,519.
Yeah, right.
Oh my gosh. It's really in a lot of new places.
One thing in the vaccination page that I found interesting was the previous smallpox vaccination does provide protection, but it may not be necessarily lifelong. And they talk about the 2003 monkeypox outbreak. And during the current monkeypox outbreak, several people who were infected with monkeypox had previously been vaccinated against smallpox decades prior. So I underlined the were infected. So, and wrote beside it, but probably protected from serious disease.
Yes.
So, it made a difference to me. Like now, just saying that they were infected in the past might have sounded to me like, oh, so there's no protection, but that's not necessarily the case.
There's a long-standing debate on how durable the smallpox vaccination is. You can detect antibodies and T cells in people decades after they've been vaccinated, but you can't do the real experiment, okay, of asking whether or not they're immune to smallpox. And I have to say that if you're somebody who is at risk for being infected by a pox virus, an orthopox virus, the recommendation is that you get revaccinated every 5 or 10 years.
Yeah, I think when I got vaccinated, they had said it would be every 10 years if I continued working with pox viruses.
Okay, so our smallpox vaccines are not likely to protect us against monkeypox. That's what you're saying.
Well, against
infection.
Yeah, they might
Disease,
not
no,
protect
disease.
us against infection, but against disease,
Probably.
I would say it's kind of I would say this is an opportunity to collect some data in that regard.
Yes. I
Okay.
think we're doing the experiment right now.
Yeah.
Well,
Yes.
the next thing we're going to talk about, there's a similar question, right? The polio
Yes.
issue
Yeah.
here. So,
Can
all
I
right.
not participate in that experiment? I'm not sure I want it.
I don't remember the consent form for this.
It was funny. The Dr. Mayo, she showed me her smallpox vaccine scar. It was big. It was like an inch in diameter. She said, "Yeah, I have a big one." I have no trouble finding it and showing it to people. It's pretty funny. All right. So Daniel Griffin likes to say monkeypox is not a gay disease. It is not an African disease. It is an infectious disease, which is really good.
Great
I
way,
like
great way of putting it.
that. Isn't that good?
Yeah.
All right. So we never talked about polio here on TWiV because it kind of happened while we were traveling, I think. And so there was a case in Rockland County some time ago in an individual who was not vaccinated. And it was, this individual initially had what was diagnosed as acute flaccid myelitis, and then they did a stool sample, and it turned out that he had polio, and this has all been extensively reported. And the sequencing of the isolate reveals that it's circulating Sabin type 2 vaccine-derived poliovirus. And of course, we do not use Sabin OPV in the U.S., so this came from elsewhere. And the sequencing says it's related to isolates from the U.K. Remember, we did talk about that on TWiV, from the UK, Sewer, and also from Israel. And so it underscores the fact that people who are being immunized with OPV, which is happening in many countries, it establishes infection in their intestines. They travel, they shed, and seems to be able to be transmitted, which is what maintains it in the sewage in the UK, that it's been in the sewage for some time. And so this individual in Rockland County has acquired it somehow. Now, they have since decided to look for poliovirus in the sewage in the U.S. Yay, we've been talking about this for years.
What an excellent idea.
So do you remember we had the SARS-CoV-2 sewer people on a couple of times? Yeah. So I emailed them all. I said, didn't we tell you to look for polio? And they said, I just ordered the primers. Because they have the samples frozen, right? They can look for it.
Yeah.
So I'm looking forward to it. But meanwhile, New York State and CDC have looked in various counties. And of course, the Sabin 2 has been found. Genetic material has been found in Rockland County, in Hudson County, Orange County. I think wherever you look around here, you're going to find it. You might even find it in Austin, Texas.
Oh, yeah. It's gonna be, yeah, for sure.
Because people travel and they bring these vaccine-derived viruses with them. But my feeling is, and Lown, you chime in, if you've been vaccinated, you're okay. If you're not vaccinated, you need to get vaccinated. Because
yes.
these someone asked me today, why is the press saying that they think hundreds of infections have happened? I said, well, because usually if you see a paralytic case, it's 1 in 100 or 200. It doesn't mean they have polio, right? They have an infection which doesn't have polio. But by all means, you know what's interesting? As soon as they found this case and they found it was polio, they set up IPV immunization stations throughout the area. And because we have so much IPV, it's not, you don't see that happening with the monkeypox vaccines, right? So because they don't have enough of that. But I think you're okay.
IPV is given routinely as part of the pediatric series, and in fact, it's in combined vaccines. My daughter got it as part of her DPT shot, I think. So it's around. Pediatricians' offices all have it, and it's being produced and it's available. So
I was shocked in these two articles that they mentioned that in Rockland County, there was a polio vaccination rate of 60.5% among 2-year-olds, and in Orange County, it was 50-something.
Wow.
Yeah. I couldn't believe that.
Yeah. So this is an area that has significant pockets of vaccine resistance and hesitancy, or whatever you want to call it. And it's for the usual reasons that we've talked about on previous episodes, none of which make much sense. But there are people who haven't gotten their kids fully vaccinated, and this is one of the ones that is often missing. And now somebody's paralyzed from polio, and that sucks.
So I have a question, because they say here that this particular virus or sequence is recognizable as something that has been found in the UK and in Israel. And I presume from that means that it's distinguishable from other type 2, Sabin type 2 vaccine circulating viruses. So does that mean that when you throw off one of these things after vaccination, that you throw off different polymorphic variants that are recognizable? So
and
you
can distinguish it from the original Sabin 2 vaccine, which was given, right? Passes through people. You have certain changes. And then as it continues to pass through people, it has a steady rate of change, and changes accumulate. And you can sort of use a molecular clock to know how long it's been circulating in people. Yeah. So sorry, go ahead.
I mean, since they're in the sewage in a couple of different places, and since we know that people who are immunocompromised can secrete this for years, and we know that these infections can occur in the community, since the inactivated vaccine, the injected vaccine, doesn't confer gut immunity, people who got the inactivated vaccine could potentially get the vaccine-derived poliovirus, have it replicate in their gut, spread it into the sewage, so you can continue propagating this virus. I would not expect it to disappear from sewage anytime soon.
No, yeah, because we're using IPV. We're getting. If you have poor hand hygiene, you're likely to get infected and contribute to the virus in the sewage. Yeah, but you're not going to get polio.
Right, you won't get polio because if you've been vaccinated. But you could still act as a carrier.
Sure.
And, and also just originally with polio, even before the age of vaccination, as Vincent pointed out, this is something that you get paralytic cases in a little under 1% of the population, but it spreads to everybody. And it's another one of those cases where asymptomatic spread is a huge driver of persistence of the virus.
In fact, in the U.S., we used to quarantine the paralyzed kids here in New York City. Completely useless because the other 99 infected kids walking around are the ones that are spreading it. So if you go to polioeradication.org, you can see that there have been many, many cases of paralytic polio throughout Africa caused by Sabin 2 vaccine. And that's because in many countries they let vaccination rates drop, and these circulating vaccine-derived viruses cause disease. And then they have to go back in. You know how they go back in and stop the outbreak? They use OPV. So they put more OPV into people because the WHO is not using an activated vaccine for its eradication. Now, one thing we have to talk about in a future episode, and we did talk about the development of new OPV type 2, right, which is genetically modified to prevent reversion. And it's now been in many, many, many kids, and there's a report in MMWR we have to go over in a couple of weeks. That should be interesting to look at that, to see if that in fact will be the solution or one of the solutions to this reversion, right?
Yeah, because the OPV is a great vaccine. I mean, it's got the downside of potentially causing paralysis, but that's extremely rare. The advantages of it are that it's extremely easy to distribute. You don't have to have needles. You don't have to have people qualified to find the deltoid and inject things. The newer versions of it are easy to transport. So it's got all these advantages for use in poor countries, which is why it's being used. But you can't stop using it. And that's something that Vincent and I argued 30 years ago and got ignored. And the problem is, as the vaccination rate tapers off, then you get these cases showing up.
It's interesting that they are using IPV in the U.S. in response to this one case, right? In many other countries, they would go in with OPV.
With OPV.
But
that's because IPV is standard in the U.S. now because, we got to the point where we can give everybody injected vaccines. And so
can you imagine the stink if they went in and vaccinated a whole bunch of people with OPV and some of them got sick?
Yeah.
Yeah.
No, that would be a problem.
Well, that was when we were using OPV, which in this country we did up until 2000, I think.
That's correct. Yeah.
There were periodic cases, you know, a kid here, a kid there, would get paralyzed by the vaccine, and that's one of the reasons we have a vaccine compensation program, which is run by the government, which exists to cover that and any other vaccine complications that are real. And that was a major argument for going to the inactivated vaccine, was do away with that risk.
Yeah, the
See,
only
this is-
polio at the time was vaccine-associated polio. There were like 9 or 10 cases a year, as I recall.
Something like that.
Yeah.
It's a bit like 1 in 2 million. So
one
however
in-
many kids
one
are
in-
getting.
1 in 1 to 2 million. Yeah.
This is reminding me I want to go back and try to figure out which of the two I had.
I think I had both.
I had both. I had one in 1955 or so when Salk first came out. Then I had Sabin in 1961-ish. I have my vaccine card actually, so I had both.
I only had OPV.
So I
Well,
think.
I mean, an incidental exposure in the lab over a period of 6 or 7 years, which probably was another dose of OPV.
I just wanted to point out that in the State Department of Health link that you gave us, Vincent, there's, I think, 2 important points. These findings underscore the critical importance of vaccination to protect all New Yorkers and New York children against polio. And you wouldn't just have to say New Yorkers, but that's New York Department of Health that's writing it. And also, they make the point that New Yorkers should know that the fact that this is related to samples found in London and Israel, this does not imply that the individual case identified in New York has travel history to Israel or the UK. So,
In fact,
Right.
he went to other countries, but not these. Yeah. No, he probably got it here from some fecal contamination, right?
right.
But anyway, there's similar questions as with smallpox, monkeypox, smallpox vaccine and monkeypox. I feel that having had polio vaccination, I'm protected against polio, not against being infected. Maybe I'm sure, actually my gut is supremely susceptible because I'm sure my mucosal anti-polio antibodies are quite low. Although on the other hand, I did work with polio for many years, so
You
maybe
worked with polio for many years, and you were a mouth pipetter for many years, too.
I was, and actually, I haven't. But I don't know when the last time I handled some polio viruses. It would be type 1 anyway, because 2 and 3 are eradicated. You can't work with them anymore. But I think that a lot of people want to know that they should get vaccinated against, and so unless you're at risk, which means to me, if you're going to countries that have a lot of polio, I don't think you need to get vaccinated.
I would say unless you know that you have some immune suppression, even.
Maybe, yeah.
I mean, if you're going to countries that have polio, I don't think I would bother to get an extra polio vaccine if I were going to visit Pakistan or something.
There's a link in one of these 2 things that Vincent gave us about should I get a polio booster now? So you could watch that if you want.
Someone in the last few weeks on our live stream said he'd had like 5 separate polio immunization. I said, why? And he said, I traveled to countries where there's a lot of polio. So, if you go to a travel doctor, they may tell you that's one thing to do.
Yeah, and if you're getting a bunch of shots for travel anyway, it might not be a bad idea.
Yeah.
It's not going to hurt. I mean, other than the needle going in.
Yeah.
Anyway, I suspect that more cities are going to start looking for polio in their sewage.
I hope so.
And we'll hear.
That'll be interesting.
Will be very interesting. I don't think every place in the U.S. is going to have it, right? There's some places that are very rural, but certainly the cities. So we'll see. And one more infectious disease. There has been 2 fatal cases of Marburg virus disease in Ghana. And this is a filovirus in the same family as Ebola viruses. And this was the first one that was found to infect people from an outbreak in a monkey facility in Marburg, Germany. I think it was 1976. And this is 2 individuals who had hemorrhagic fever. They went to the hospital and they both died within days.
There's been a 3rd one.
There's a third one as well.
Yeah, it's a child. And,
Oh my gosh.
The dead child, whose gender or age were not disclosed, was one of 2 new cases reported last week by WHO. Of the 2 cases, one is the wife of the index case, and the other is the child of the index case, and the child is the one who died. The wife is still alive.
It's too bad. So this is a virus whose reservoir is known to be the Egyptian fruit bat in many of these African countries. In fact, John Towner was at the bat meeting that Brianne and I went to. I was talking to him, and he said, Ebola, we're not sure what the reservoir, but there's no doubt that Marburg is in Egyptian fruit bats, and people, you can track them and see where they go out at night and they feed. They're fruit bats. They look for fruit trees, and if there are people around, they can infect them. And in fact, Jonathan Towner, by the way, is the guy at CDC who crawls into caves throughout Africa and gets bats. And I said, would you like to come on TWiV and talk about it? He said, yeah. He said, in 2 months, because I'm going to Africa for 2 months. So we can hear him talk about it.
Nice. So, reading through all of this, all I could think about was the bat meeting. Mm-hmm. And some of the things that we heard from Raina Plowright, particularly in her talk, but also when she was on TWiV, about things that make spillovers more likely. Yeah. And so immediately I started thinking, well, what's going on in Ghana that could be leading to, increased numbers of spillovers? So
there was a talk, and I have notes to get a lot of those people from the bat meeting onto TWiV to talk about their work. It's very interesting, including in that last one where they're treating rabies with a monoclonal. That was just an amazing talk.
I'm sad I missed it.
Oh, yes, right. You had left. But anyway, so there's a guy from CDC. They go to, I think he goes to Uganda, and he puts trackers on bats. They capture them, and they put trackers on.
This was so cool.
And they can then use Google Earth to see them. And they said these things fall off. They use surgical glue, so they fall off in a couple of days. But they can see, and they go out at night. Every night, they fly 40 kilometers to the same fruit tree. Right? It's
in somebody's backyard.
In somebody's backyard, right? And then they fly back, 80 kilometers round trip. Amazing. They can go back to their cave, and then the next night they do the same thing. So you could see where, if they're in a burst of virus shedding, which is what happens, these bats have bursts. Maybe it's triggered by hormones or food short or whatever. And then they could be shedding, and then someone would get infected. So really cool stuff. That's why I put this in here, because the Marburg reservoir is quite known, and clearly bats. So
I'm looking at for anybody who wants to look it up, the wiki page on Marburg virus, and it looks quite good. Among other things, it summarizes all the outbreaks that have gone on in the past.
And the other thing we heard at this meeting, people now, they get bats. You can either breed them or capture them. You can infect them with Ebola and Marburg, and they're fine. The bats are fine. They don't get sick. They shed virus for a while. I don't remember. Do they make antibodies, Brianne? I don't even remember if they make antibodies.
I think they made antibodies, and maybe they, had a slight body temperature change for maybe a day. But in terms of their activity, there was no
one
change.
Lady, Silke, who visited the incubator. She works at a BSL-4 in Berlin. They shipped bats from Uganda and infected them at the BSL-4 in Berlin. And she said, we can't repeat this experiment because the airlines don't let us ship bats anymore. They put an end to it. So, fascinating meeting. Okay, and maybe the best thing I got out of it, Brianne, is that people like Raina Plowright think being reactive is too late. You should prevent the spillovers by trying to prevent the ecological disruptions that we're doing. She said, for example, in Australia, you could plant eucalyptus trees, and the bats would use those and not go look for fruit and, horse farms and so forth.
The fact that there were such concrete ideas like that,
that's great.
Where they had the data that basically showed that the spillovers were partially related to a lack of eucalyptus trees for the bats to go to, so they were going to human habitat. And it was fascinating.
That's why this meeting was great, because it was really multidisciplinary. And, as a virologist, I think about the viruses, but not much about the ecology. And there were a lot of ecologists there who say, in fact, someone asked a question. He said, what does all this sampling do? You can't prevent it. You need to deal with the ecology, right?
Well, it makes me think of SARS-CoV-2, right?
Yeah.
And SARS-1. All right.
Yeah, for sure.
Those are cultural behavioral issues, right?
Yeah, for sure.
The markets.
Oh, by the way, I have to tell you guys that I was sitting in the plane on the way to the meeting, in the back, and somebody walks up to me and says, "You're the last person I would think not wearing a mask." And I look up, and it's Peter Daszak. Was he wearing a mask? He was wearing a mask. Yes.
Yeah, okay.
He sat down next to me, and we chatted for a while. So we're gonna. He told me I had several meals with him, as you did too, Brianne.
I did.
He has a lot of.
Did you wear masks?
It's interesting. No,
Not while eating.
Not while eating. No.
Yeah.
So in at the university, it's only suggested. There's no masking requirement, and some people did, and some people didn't. Yeah.
Kathy, actually, we're going down a rabbit hole here, but I want to know: do you have any feedback on virus at the meeting, the ASV meeting?
Mostly anecdotal, nothing real data. And then we had more than several people write or muse, should ASV survey, or should we send warnings out to people who are at the meeting? And it's kind of like, well, survey, we're not going to go there with IRBs and so forth and so on. And these are all virologists. And if they can't figure out that if they have symptoms, what they should do, I mean, we spelled it out in all the meeting materials.
Yeah.
But
I'm just interested in how much transmission there might have been, because
and
I thought the cool part was you asked people to wear masks, and they did.
Yeah, until you got to the dance toward the end of the evening. But and so there were reports of half the people in this lab and half the people in that lab, and in contrast, I know of somebody who was extremely careful, always wore a mask except when eating and drinking, and came down with symptoms and was positive after they got home.
Yeah.
So. Yeah, it's like I said all along. If you think the virus isn't going to be there, you're crazy.
Hey, Kathy, here's an interesting anecdote for you. So Tony Shouts, right? He's the meeting organizer. First day, he says, well, we have to recommend that you wear masks. You don't need to, but we have to recommend it. The university requires us to do that. And he said, after all, we don't want to have a super spreader event like ASV was.
Yeah. Well, but then, I'm still on the no COVID island. I don't know about
Me too.
Rich, Brianne, and Vincent.
Me too.
Yeah.
So
I'm amazed. I was there for 5 days with 1,700 people.
Yeah.
And sometimes crowded in unmasked conditions, like the banquet and the meals.
Yeah.
And you got to wonder: is this certainly probability is part of it, but
But the other possibility is that we're mutants.
Yeah, we're
so
mutants.
Somebody has
That's right.
proposed that to me.
Sequence your genome.
I've been on how many flights this summer? How much travel did I do in July? Many known exposures in July, and
yeah.
Well, I have to say,
knock on wood.
I was in Key West last December, and then my biggest adventure was going to a Bruins game, and then coming back on the T, with all the Bruins fans. I mean, forget it.
So I mostly don't wear masks unless asked, but I was on the shuttle bus from Fort Collins, Brianne, and it's a small bus with 9 people. Mm-hmm. And all of a sudden, someone behind me starts hacking away. I whipped out my mask. I said, you may not have COVID. Maybe you have something else. I don't want it. Yeah. So I think I'm going to carry a mask forever. And when someone's hacking around me, I'm going to wear it.
Yeah, I wore one throughout all of the travel, but I think part of it was that I wanted to make it through all the travel without getting sick.
Yeah, that's true. If you have another meeting next week, you don't want to miss it. Yeah.
Yeah.
All right. We do have 2 papers for you. And we will get through them. It's not a problem. The first one.
Those were the short parts of the show for the first hour.
Yeah. The first one is a snippet, but I don't. As someone said, stop calling them snippets. You don't really do that. But it's a Science Advances article called "Ancient Herpes Simplex 1 Genomes Reveal Recent Viral Structure in Eurasia." This is really interesting. article. This is from many different universities in very different places, like the University of Tartu in Estonia, University College London, Cambridge, Leicester, Aberdeen, Alan Turing Institute, wow, and many, many more. The first two authors are Miriam Guelil and Lucy Van Dorp, and the last two are Charlotte Holdcroft and Christiana Scheib. And so this is all about ancient DNA and what you can do with it. And, you know, you have ancient human DNA, you have ancient viral DNA, and you can start to know, hopefully, when certain viruses appeared in people, right? Because they came from animals at some point. And they make the point in the introduction, you know, co-analysis of host and pathogens can give you interesting information, but they're often challenged by, you know, mostly we have recent sampling, right? Recent people and recent viruses. And they say herpes viruses are an example of this. We have not many ancient genomes, and we make a lot of assumptions about when it arose. So they basically tried to get some ancient DNA from people and do some sequencing. And so the herpes simplex virus is an interesting one, as you know, double-stranded DNA virus that infects many, many people in the world. They say 2/3.
Most of us.
Most of us. They say at least
That's
2/3,
most of
but
us, yeah.
I think it's most of us, yeah. But interesting.
They say here 60%, but you know, my teaching was always seropositivity in excess of 80%.
Yeah. And so, interestingly, there are few genomes of herpes viruses.
For the newbies out there, think cold sores.
Yeah.
Okay.
Indeed. Indeed. And that will make an interesting connection with cold sores at the end of this,
Yes.
which is very interesting. And so from the existing genomes, there are 3 phylo groups, right? 1 Europe and America, 2 Europe, Asia, and America, and 3 Africa. And they think this group 3 is basal to all the others. So they think that the viruses, the diversification of the different groups coincided with human migrations out of Africa. But there are more recent studies which say, no, that's not right. Herpes is more recent. And the most recent ancestor is less than 7,000 years ago. But the problem is most of the genomes we have are from recent clinical isolates and highly cosmopolitan centers, and the oldest genome is from someone in New York from 1925.
Right.
And they
So
say
to make
even
statements,
that.
statements about the evolution, you're doing huge extrapolations
Yes.
based on
this
comparison.
This one in 1925, they say it was passaged for generations before the genome was sequenced. So
Yeah.
you need something fresh.
Yeah, and to be clear, herpes viruses in general infect everything. I mean, they're like all the primates have herpes viruses, and the fish have herpes viruses, and the corals have herpes viruses. So, but the specific question here is, where did the strains that we see in humans, where did they come from? And there's just been this
sort of
void of data.
Were they with us when we were primates?
Right. When we came out of Africa, were they with us when we first became humans, or did this set condense down
emerge
at some point, or emerge at some point and become much more prevalent?
So that's not to say that we're not primates.
So where do you get ancient DNA? Well, they said you can find herpes DNA from
you dig up the bodies.
Teeth.
Oh, but specifically,
from teeth. So there have been previous modern recoveries of herpes from teeth, subgingival plaque of living people. You can also find it in the trigeminal ganglia, where the virus goes latent in cadavers. So they said we should be able to find ancient genomes. So they went looking. They found 4 ancient DNA libraries from teeth that had herpes simplex 1 DNA sequences. They came from a number of people. One was a young adult male from an urban medieval hospital cemetery, 1350 to 1450 CE, and an adult female from an early Anglo-Saxon cemetery. So these were obviously exhumed. 500 to 575 CE in Cambridgeshire, UK, an adult male from a burial related to the Nevellino culture, 253 to 530 CE, and that's in Russia, and an adult male from the Netherlands.
Now it's in Russia. We don't know what it was called back then.
Yeah, 253 to 530. Who knows? And then from the Netherlands, 1600 to 1700 CE. So they have
these
libraries.
extend from the Bronze Age to the like 1700.
Yeah.
So it covers a pretty broad stretch of history.
This whole ancient DNA thing just boggles the mind. It's amazing to me that you can do this.
Yeah. So the Bronze Age, I had to look this up, 3300 to 1200. All right, so tell me, what has replaced BC? Is it CE?
So CE is Common Era. That's replacing AD.
Yeah.
And BC is now BCE before Common Era.
Before Common Era.
Okay, because Wikipedia still has BC. I don't know. They need to change it all, I guess. Okay, BCE, 3300 to 1200 BCE is the Bronze Age, and that's going to be important.
No, I think it's 1200 CE.
To 1200 CE.
Oh, CE goes past.
Right. It's a long period of time before we figured out how to smelt things other than bronze, basically.
I love history. So anyway, there were libraries that they found from these individuals, and they did some genome sequencing, and they found herpes sequences in them. And they did a number of really interesting analyses. The first thing they asked is whether these individuals had any of the known susceptibility polymorphisms, because they say, well, maybe if they were particularly susceptible, that would explain why we can get herpes virus out of them.
So I think one of the questions is that if you look at all of the sort of ancient DNA samples that are out there, the prevalence of herpes sequences in them is pretty small relative to what you would expect in terms of herpes distribution. So the question becomes, did these people have something special about them?
Right.
And that's part of this question. And there are other possible explanations for that as well that we might get to.
Right. Well, one of the explanations is this is a virus that goes latent and is not going to be obvious. It's not like circulating in the blood all the time. It's holed up at the trigeminal ganglion. Right. And with as the bodies decompose, the ganglion, that's nervous tissue, that's going to decay. So this finding of teeth, sequencing the pulp inside the teeth, is kind of, I think, the unique approach here. And I think that may be why they managed to find it. But yeah, they had to ask, were these people who were particularly susceptible, is that going to skew things?
And
one of the things is that the root of the molars that they were looking at is very close to the trigeminal ganglia where the latency happens. You just made me flash on something, Alan, because they talked about the reactivation around the time of death. And it makes me think that in experimental systems, I'm really going off the wall here. In experimental systems, the way you react, one way to reactivate virus, is to take the trigeminal ganglia out of the animal and stick it in a dish. So it's no longer in its normal immune context. I wonder if that's the same thing as dying, right?
Yeah.
Yeah, it's a good point.
That's an idea.
Yeah. I'm not finding out for you.
Okay. I'm not.
That's a good point, because they mentioned that it's been found in viral DNA has been found in trigeminal ganglia of cadavers, indicating it can reactivate peri or post-mortem. Yeah.
Yeah, right.
So, yeah, interesting. Anyway, they don't find the variants that are known to lead to more susceptibility, except one person had an HLA-B haplotype, but the others didn't. So they rule that out. They then said, okay, so did they have some kind of a lifestyle that stressed them? I have a feeling that back then everybody was stressed, right?
Yes.
Yeah.
Life was tough, right? And they found, this is great, they can look at the isotope data from the tooth and bones and find out that they had a diet low in animal or marine proteins, perhaps due to poverty and an inability to take in sufficient nutrition. So that
applied to
JSD,
one of them,
one
right?
of them.
So this was the one found at the Hospital of St. John, which was a hospital for the poor, so it kind of fits. The guy was probably poor.
Then they say, herpes 1 is found in higher prevalence in people with chronic and aggressive periodontitis. And so they said two of these individuals have dental pathology consistent with periodontal disease, and one of them was a tobacco smoker. Oh my gosh, you can figure this out. Isn't that amazing?
And well, if you look at the supplemental data, because I read that, was like, how do you know? And you go into the supplemental data, and they've got a photo of the skull, and there's the teeth are worn in a way that's distinctive of having held a clay pipe frequently in their teeth in the same spot, and the clay is abrasive, so it wears away.
That's the way people smoked back then, with a pipe, right?
That's the way people smoked.
Because you couldn't
I
just
guess
buy
they weren't smoking Marlboros.
They weren't
right?
smoking Marlboros.
Yeah, so this guy was going around with a clay pipe clenched in his
That's so
teeth
cool.
over on one particular side, and it wore away.
Which they say, tobacco smoking is a major risk factor for periodontitis. They find bacterial sequences that are, consistent with periodontal pathogens as well. So that's another thing there that they find. Then they had cavities also, and you can see that a gross carious lesion in the upper right second molar, and one of them had an abscess that perforated through the jaw. Oh my gosh! Because people didn't get.
That must have hurt.
There was no dental care back then, right?
Yeah.
No dentist. So you had cavity. It kept going until they pulled their tooth out, I guess. I don't know. Anyway, let's see. They conclude that it's not possible to distinguish between, in these cadavers, primary infection, reactivation, or rare immune defects. So they say these infections are probably typically reactivated infections, although they could have had some other issues as well. Okay, so
I want to make sure this is clear for the newbies.
Yep.
Okay, very briefly, you get infected with this particular herpes virus type 1, in the mouth. You can get a primary disease that's like blisters and stuff that's really cold sores gone nuts, or you can get nothing. Most of the infections are asymptomatic. And then it goes latent, which means that it, in this particular case, goes to neurons in the trigeminal ganglia. The virus is not actively replicating, but the genome is there, and it's maintained for the rest of your life. And occasionally, under situations of stress, psychological, physiological, that I think usually impacts on your immunity, I think that's the reason for this. The virus is probably always just ticking out a little bit of virus, but if your immune system sort of checks out for a few days, the virus can get an advantage and then reemerge once again as cold sores. This is why people get cold sores recurring, okay? But the genome's there forever. So that's what we call reactivation. So there's a primary infection, there's latency, there's reactivation.
Right.
So they can't distinguish between a primary infection, reactivation in these individuals, basically. Yeah. But if they're in their, I don't know what the ages were, but they're not kids, so they probably have been already infected.
Yeah, they don't
previously.
have exact ages, except one appears to be age 17 to 25, I guess, based on skeletal structures. The woman is, they say, middle age because she had signs of beginning arthritis, and then the others are just adult.
Yeah.
So some age range in there.
And the last thing they did is ask, well, are there any genome changes that we know are associated with different kinds of pathogenicity? And they don't find anything that would say, there's a change compared to what we're seeing today. But I think that's really hard because they say we do not detect large-scale changes in the genome, but you wouldn't need a large-scale change, right, to make a difference in pathogenicity. So I'm not sure how much you can get from that. All right. Anyway, so then they take these three genomes. They get three full genomes of HSV1. It's really good, and then they mix it with all the known genomes that they have today from virus isolates from Europe, Central East Asia, Africa, the Americas. And
right. So they got three full genomes, and one of the individuals, the DNA was apparently degraded, and they can only get partial bits and pieces. So that was excluded from most, but not all of the analyses.
And so the rest is computational biology to try and get an idea of the relatedness and when these might have arisen. So they say the two oldest genomes are basal to phylogroup 1, which it means they're ancestral to phylogroup 1. And the medieval genome of theirs is in phylogroup 2, which is closest to herpes sampled in Germany and Finland. But the most interesting, of course, and they have a lovely phylogenetic tree which has dates on the bottom, right, which has a 0 there, which is transition between BCE and CE now, I understand. Hopefully I can remember that. It goes back to minus 4,000 and 2,000, which is where we are today. And they can place their viruses in that. And so the most recent common ancestor of all of these herpes viruses, and they use the measurement Ka, kilo annum, right, 1,000 years, is about 4.68 kilo annums. That 4,680 years ago, right, with a range of 3.78 to 5.65. And so this is I think the new date for the origin of herpes simplex 1. Now, this corresponds to so the key here, and where they try and make it more digestible. So they say our results are consistent with either a lineage replacement or a relatively recent emergence of extant HSV strains. So, everyone has herpes. So the idea is that it left Africa with us, and then it diversified. And they say, an interesting discussion, they say it could be due to increased transmissibility, but they say we don't see any changes in the genome. So they say, we don't think it's the genome. We think it's human behavior that
it's the people
that changed the pattern of transmission. There were migrations all over the place. There were migrations from Eastern Europe into the rest of Europe, and increases in population density during the Bronze Age. And they say the primary mode of transmission is vertical, parent to child, and the addition of lateral transmission as population density increased during the Bronze Age, potentially linked to the introduction of new cultural practices such as the advent of sexual romantic kissing, may have contributed to a shift in the dominant lineages which have continued to circulate this day. So in other words, all these contemporary HSV ones are derived from something ancestral to these, and there was a big shift around the Bronze Age, and they're speculating that this might, the population density and maybe kissing might be it.
So as to the kissing part, in the sentence that Vincent read, there is a section that there's a parenthetical about going to the supplemental materials.
Yes.
So I had to look at the supplemental materials, and there is a section in the supplementary text called A Brief History of Kissing. And it's quite the history. And not something I ever expected to read in a scientific paper, but I got a big kick out of that.
Yes.
It comes from a book called
I want to correct something I said earlier. The Bronze Age actually did end in 1200 BC. So it actually none of these bodies are from the Bronze Age, but the
BCE
origin point of herpes virus goes back to the Bronze Age.
The Bronze Age.
Apparently kissing as well.
Of the extant strains, and kissing apparently goes back to the Bronze Age.
So, Brianne, I even tried to dig into their primary data on this because they reference some Neolithic figurines, and they reference a manuscript, and that reference 112. I went back, tried to dig up some of that,
which they call the earliest known record of kissing.
Right.
Yeah, and but I think that
the
reference
dawn of porn.
That reference must not be right because it's the figures don't line up and stuff. But in trying to sort this out, one of the references was 111, which is the author is A. Rose, thesis, University of Cambridge. Okay, and that's the one. That's the reference about, let's see here. Where's the brief history of kissing again? Neolithic figurines interpreted as a couple embracing. Okay. A. Rose is one of the authors on the paper. Okay? So I wonder what, it's Alexa, I think. I wonder what her thesis
Alice.
is. It's her, Alice.
Alice
Is her
Rose.
thesis about kissing? I don't know.
I had no idea. I just assumed people always kissed, but
Yeah.
Does
So
anyone know?
Yeah, this is news to me.
Do non-human primates kiss? Does anyone know?
So to
Bonobo.
jump ahead, it's
Yes.
in
According to this.
it's in this
So,
paragraph.
yes,
Let me read you the second paragraph. This is by Christiana Scheib, one of the authors, and this second paragraph derives from a book called The History of the Kiss by M. Danesi. Okay, such a transition in transmission routes would allow for transmission within larger host communities as well as at the intersection of populations when they meet. Sexual romantic kissing is far from universal in human cultures and is also observed in chimpanzees and bonobos, but is higher in prevalence in modern European, Middle Eastern, and Asian cultures. So
Interesting.
that answers your
There you
question
go.
about primates.
Yes. Also, in the previous paragraph, apparently an emperor at one point tried to ban kissing in order to stop the spread of disease.
all people. Yes,
Interesting.
Tiberius, who I think probably died of neural syphilis or something. But,
Yes. They note that it is unclear whether the disease was herpes.
yes.
But
I'm always interested in things like that that imply that people understood contagion.
yeah.
Yes.
But well before germ theory and at a time when they were still talking about miasma
For sure.
stuff.
I think they did. I think many people did, right? But it's really interesting. It's always about the people in the end, right? I mean,
Yes.
the things we do like this, and also what we talked about in terms of spillovers and
Yep.
bats. It's just,
And
Amazing.
monkeypox.
And polio. Yep.
and so you don't blame the bats or the pigs or the birds, folks. You just blame us. It's our fault.
Yes. Yep.
Anyway, I thought that was fun. And
Yeah,
actually, I found a
really good.
Philadelphia Inquirer article on
Yes.
this where they talk about the kissing hypothesis. And they speak with Christina Scheib on this. It's really interesting.
That's a very good article.
Did I put it in the show notes?
I think, you linked
Yeah,
it.
you did.
Oh, yeah, there it is. Philly Voice. Yeah. Oh, there's a quote from Christiana Scheib. Yeah. So every primate species has a form of herpes. We assume it's been with us since our own species left Africa. However, something happened around 5,000 years ago that allowed one strain to overtake all others, possibly an increase in transmission, which could have been linked to kissing. Very cool.
Yeah, that's a really nice summary.
I like that. All right. And we have one more article, which we promise we will get through expeditiously. This is a Cell Reports Medicine.
It could
Well,
be
they-
a snippet.
This could actually be
Yeah,
a
I know.
Yeah, this
Because
is just
This is our
Basically-
snippet.
Yeah,
This is our snippet, sure.
So here's the answer. So they look at different vaccines. What do you give as the second boost or second dose after you give the first one? And this is because of a lot of countries don't have enough vaccines. So what happens if you mix and match them? And the answer is that you will get a good response, particularly if you give an mRNA vaccine as the second dose.
Right.
And that mixing is almost always better than the original homologous first and second dose the same.
And the last sentence in limitations of this study, moreover, we only evaluated the antibody response induced by vaccination, not the response mediated by T cells.
Right.
Or
Yes.
disease,
Right.
right.
That would be good
No,
too.
they just looking at antibody
Yeah.
response and
Right.
yeah.
And they look very broadly at a lot of different vaccines,
Yes.
not just kind of the usual suspects of the mRNA
Yeah.
vaccines and the adenovirus vaccines. In fact, I had to spend some time making sure I was clear what BBBIBP Corv
Yeah.
it was.
Oh, I just
Yeah,
looked at the
that's-
picture and the graphical abstract. And it's a
This,
busted
I
up
thought-
coronavirus.
Many people ask, "Oh, I live in this country. I've had this," So I thought this would be of interest to people, and in particular, a lot of people who've gotten the adeno-vectored vaccine who don't want to get a second dose, right? Because they're afraid of the side effects. So they ask, "Which one should I get?" So I thought we
Get the mRNA.
So this is immunogenicity and reactogenicity of heterologous immunization against SARS-CoV-2 using Sputnik. Is that a V or a 5? I forget.
That's a V.
I think it's a V.
Chadox 1S, BBIBP,
Corvi,
Ad5 nCoV, and mRNA 1273. And this is from a big group in Argentina, and I wanted to point out there are 5 co-first authors: Carla Pasquale, Augusto Verese, Diego Ojeda, Marina Pasinovich, and Laura Lopez. And then we have the corresponding author is Andrea Gamarnik, who I know from Polio days. She used to be a postdoc with Raul Andino, and I met her way back then. And then she went down South America to do open her lab, worked on dengue, and now she has joined the SARS-CoV-2 fray with this work. So really very nice. And as you've heard, this is to try and see what would work as a second dose when you've got something that's either you don't want to get it again or is in too short supply. And in Argentina, they have a lot of different vaccines. So what is their list here? They have
Sputnik.
They have Sputnik,
which is the Gamaleya Institute's the Russian vaccine. It's a recombinant adenovirus. It's actually 2 recombinant adenoviruses. The first immunization is with a Ad26 vector, and the second is with an Ad5 vector. Then they have another Chinese adeno 5 vectored vaccine called Ad5 nCoV.
Right.
Then they have Chadox, which is the chimp adenovirus vector vaccine. They have this BBIBP, which is inactivated coronavirus, also from China, Sinopharm. And then they have both mRNA vaccines, so they only really report on one of them. That is Moderna and Pfizer.
Yeah, so they're using all of these in Argentina, which makes it a good place to do this study. So it's basically they went across Argentina. They got 1,300 people who got a first dose with these various vaccines: Sputnik, Chadox, BBIBP, and then they asked, what did they get next, and how did they respond?
And over 90% of Argentinians over 50 have been vaccinated with 2 doses.
And that's telling, because that's their whole idea here in allowing the heterologous vaccination was to get better coverage. Yeah, and from that point of view, it's successful.
Yeah, and so this provided a natural experiment. I mean, this was not something where they set up and said, okay, this group will get this one first and this one. It was just, okay, we got all these different vaccines and you can get them whichever way. And then they went and they found people and saw what order they were in.
So is that, I was trying to decide whether they did that so it was not at all blinded and it was
I don't think
all
so.
post hoc.
I think this is just, I think this is all post hoc.
Yeah, these are just vaccines that people got, Yeah,
so they enrolled people who had received a first dose of...
Oh, no, it wasn't post-hoc. They enrolled people who got 1 dose.
Oh, no, wait.
And then they gave
that's
them...
what I was thinking.
Right.
Yeah, you're right, Kathy.
I'm sorry.
You're right, Kathy.
I stand corrected.
It is. So they had a first dose, and then they divided them into different arms.
Divided them into groups.
And
Okay,
gave them different
that's right.
second.
Right.
But what I didn't see anywhere, and I just did a search of the PDF for the word blind. I don't think it was necessarily blinded. So the recipients may have known what they got. And I wondered how that affected their reporting on reactogenicity, the side effects that they got. But I was just curious overall whether they knew which they got for their second dose.
Right.
Yeah, the mRNA gave the most reactogenicity as the second dose, right? But they did follow that up. They looked at, adverse effects. As you would expect, pain at the injection site was the most frequent. No serious adverse effects, hospitalizations, or death in the 7 days after the second dose. And then they took serum from these individuals and measured anti-spike IgG, and they did some neutralization assays as well. And they look at the antibodies 14 and 28 days after the second dose. And so here are the results for Sputnik. The combination with BBIBP or Sputnik itself was inferior to that of Sputnik C1, C2. Remember, they had 2 different vectored Sputniks, right?
Right.
So that was better than giving Sputnik C1, or BBIBP. But the combination with mRNA, ChAdOx or Ad5 nCoV was equivalent or superior compared with the homologous regimen. The ChAdOx cohort showed that the heterologous schedule was with BBIBP was inferior compared with the homologous scheme. So if you got ChAdOx, that was better than getting ChAdOx BBIBP. The use of either Sputnik C1 Ad-nCoV or mRNA 1273 gave equivalent or superior responses. In the BBIBP cohort, where they first got that, combination with Sputnik, ChAdOx, or mRNA was superior than getting a second dose of BBIBP. For all cohorts, the highest response was induced by mRNA as the second dose. Gee, there's something about that, huh?
Yeah.
That
I'll
mRNA.
tell you, mRNA vaccines are really hot.
Yep.
Or at least the mRNA vaccine for this particular pathogen
For this particular, yes.
is really hot.
So this is a question that came up repeatedly at the height of the pandemic, where
Yeah,
people would
yeah.
write in and say, I got a first dose of this, and should I get a second dose of the same thing? And our usual take on it was get what you can get.
Yeah.
Basically. This looks like mix and match is okay. And if you have a choice, get the mRNA second. Although I don't know how many people there are left who are going to get the vaccine who haven't gotten any.
Well,
But if
that's
you're
in
out
the
there-
U.S.
Yeah.
We're still talking about in a lot of countries where they might only
yes,
have
there are
one.
a lot of folks where they might want to, a lot of places where they might want to implement something like this.
They
And
also
as you
looked-
pointed out, this is based on binding antibodies and neutralizing
Yes.
antibodies,
Yeah.
and not
Yes.
protection from infection.
so all these
There
neutrals.
is no downside
Right.
from
No,
what we can
there
see
isn't.
here.
There's
Okay,
no downside,
there's
yeah.
no downside in terms of adverse effects. There's no suppression of the immune response or anything
Right.
like that.
No. Yeah, in fact, Daniel, on the latest clinical update, said as much here in the U.S. If you get whatever you can, they're all fine. They also looked, so the viruses that they use, the spike that they use for these assays were the ancestral homologous, but they also tried BA1, Omicron BA1, and they find low neutralizing titers in all arms of the study, as you would expect, because that's what we know. And they say a large proportion of samples in the Sputnik, ChAdOx, BBIBP cohorts were negative for the presence of neutralizing antibodies against Omicron. But in all three cohorts of these three, mRNA vaccination as the second dose gave you an increase in serum neutralizing titers against BA1 compared with the homologous schemes. And that's something we sort of know, right? A boost broadens your antibody responses. And
Right, so
their timing was
even though
interesting. Go ahead.
I was
go
just saying, even though these aren't that good against the Omicron, if you get the second dose with the mRNA, you might
Yeah.
have a better antibody response.
Yeah. I was going to say their dosing timing. The time intervals, according to the definition in Argentina, at least 8 weeks for viral vector vaccines and 4 weeks with an activated vaccine. So that's the timing between doses. It's
And
interesting.
I wondered what sort of impact that could have, because as we suspect, at least, one of the reasons the boosters are good is that you've had a long interval between your primary series and getting boosted.
Right.
To
Right.
sort of incubate. So there's at least a 1 month difference in time. I don't know if that makes
Yeah.
a difference. It was
Yeah.
also not clear to me. They talked about the interval, whether they were talking about vectored or inactivated vaccines. I don't know. I presume they're calling the mRNA vaccine not, I don't know what they would call that.
Right.
No? Is that
It's
vectored,
not. It's
or
not
is it
inactivated,
inactivated?
right?
No.
It's something
it's
in
not
between.
really vectored, except it is actually being synthesized in you.
Yeah,
Okay, so it's closer to vectored than something else.
I think so. Yeah. So they make a very interesting statement in the discussion. They say, and they say the booster strategy is on its way in most high-income countries. It's safe, prevents severe disease. However, many low and middle-income countries in Africa, parts of Asia, Eastern Europe, Latin America are still unable to implement this strategy. And they say our observations could provide valuable information to decide the best combination of vaccines to apply in heterologous systems for prime vaccination as well as for boosters. I think that's an important point to remember. We're arguing about booster 1 and booster 2 here in the U.S. and in many of these countries, they can't do that yet. They're still trying to get the second dose. So,
and it's apparent that first boost, at least, has an effect, an important effect
Very
against
important,
Omicron.
Seems to have a big effect.
huge, yeah. Yep. All right.
God, what a journey. This whole thing. What a journey.
Yeah.
I'm kind of in a shock still, I don't. Did we do this for 2 years? Is that right?
It's
Oh yeah, there's a gap in my life another. I went to Mars for a couple of years,
I look back at some of the TWiVs we did, and I don't remember doing that one. That's a pretty cool title.
So
the world has been divided into a before and an after now. Yeah.
I don't know.
BCC and
Yes.
There's
NBC.
one of the messages here that I don't know if we really emphasized is that this particular inactivated vaccine doesn't look so hot. Okay.
No.
And I think we kind of knew that, but this reinforces that. That doesn't mean that all inactivated vaccines, or even all inactivated vaccines against this virus, are going to be inferior. I don't think we really have the data, but this particular one, at least, is not so hot.
They also mentioned that they have a similar result with another inactivated virus.
Yeah.
Okay.
Or that one was published or something, yeah.
So
when I look at that, I think the confirmation of the spike is important. Okay. Mm-hmm. And these inactivated viruses are not going to have any stabilizing mutations in the spike. And we already know that some of the inactivated agents, like propiolactone, can trash the spike. So you wonder if for this particular vaccine, if the inactivated, and this is always a problem in inactivated vaccines, is whether the inactivation technique is going to trash the antigen and trash the immunogenicity. That's what I think of when I think about this.
Right.
now, of course, for polio, it does not. It's very good. IPV is a great vaccine. But obviously, it's not the same for all. So China has depended on these two inactivated vaccines, and they haven't done so well. I just found an article in Nature from the end of June saying that they have made mRNA vaccines and they're doing human clinical trials. And it says, in a small clinical trial, the Chinese vaccine candidate mRNA triggered a stronger antibody response in adults when given as a booster than did a jab containing inactivated vaccine, the platform the country has mostly relied on so far. So that's very similar to the results in this paper, right?
Yep.
Yeah.
Give a second. So they could solve their problems by just boosting everyone with mRNA vaccines. Yeah. By the way, I heard that George Gao stepped down from the CDC in China.
really?
Yeah.
Oh, is
Yeah,
that
I heard
right?
that too. Yeah.
So it was a tidbit from Peter. And Andrea Gamarnik was a plenary speaker, ASV 2017. Where was that held, Kathy?
Madison.
Madison. Okay, I probably
I went
saw
to that one.
Is that the one where we did a TWiV
was-
with all of us?
Yeah. And Mavis.
Yeah, that was sponsored by Promega, right? Very cool. All right, let's do some picks, folks, so we don't keep your ear for too long. Brianne, what do you have for us today?
So I have an article that was recently published in The Atlantic by another Jeopardy contestant from this summer called Everyone Loses on Jeopardy Eventually. And she is sort of chronicling her experience. She lost in her first game. But in general, I would say that this article describes my experience extremely well. Both kind of the excitement of going, the excitement combined with mortification of having everyone watch you on TV. It was sort of this weird combination. And the extreme fun of getting to hear everyone's responses and hear about the community of people watching after the fact. The number of messages I have received from people throughout my life who've told me about watching with their kids or, getting together with other groups and watching together, and how much that has been an unexpected, huge benefit as has been becoming friends with all of my fellow contestants who taped on the same day that I did, were described pretty well by this article. So I've been sharing it with everyone I can.
Oh, this is great.
Brianne, you have experience with being on camera in front of a lot of people.
Yes.
Was this scarier? Or scary at all?
I was very nervous the entire time. Extremely, extremely nervous the entire time.
Of course, you're being tested too. I mean,
you're being tested.
Yeah.
You're being tested,
in a competition.
and it's a thing that I, imagined doing since I was a little kid.
Okay.
So there was a lot of, wait, is this really happening?
Yeah.
So this is very cool. The only way to make it onto the show is to pass 2 different online tests and a practice game on Zoom. The producers told us that more than 100,000 people took the initial online test for this in 2021, and 99.6% of people who take the first test that year do not make it. The same year, Harvard rejects 96.6% of applicants. So it's tougher to get on Jeopardy than to get into Harvard.
Yeah,
That's
so
very cool.
I like I said, this sums up a lot of pieces of my experience really well.
So, Brianne, you said that people had heard of TWiV on Jeopardy, some of the contestants,
yeah,
right?
so there were 12 of us who were together for the day, and 2 of the 12 were TWiV listeners.
Okay, so
That's
is
amazing.
our percentage of the U.S. population that knows.
Not so sure.
You think it's lower?
Remember, these are
We reach a sixth of the country.
these are trivia freaks. Remember?
Yeah.
Yes.
Yeah, it's
Okay.
true. It's not a
So
good sample. Yeah,
I think we're a great source of trivia.
very cool. You can't do it again, can you, Brianne? This once is it, right?
No, it's you get to do it only once unless they invite you back for some sort of tournament or something like that, which I would do again in a heartbeat. But really, I can't do it again.
Okay.
Wow.
Very good. Kathy, what do you have for us?
I have way too many windows open now. Where is the Google Doc? Oh, it's the one. Got it. I chose a climate spiral, which is a short YouTube video that just, when you start watching, you should think, how big are those circles going to go? And then it turns it up at the end. And then the next follow-on YouTube for me was Carl Sagan talking about climate change, which I have to admit I haven't watched. So it's not a valid pick from that standpoint, but I didn't have time. I meant to. So, but the climate spiral is just really kind of sobering.
It is dramatic and
very
well
good.
done.
And I love the
Cool.
watch it till the end, dudes. Don't bail out because it goes through a transformation at the end that's very good. And I didn't watch the whole Carl Sagan thing either, but it's important that this is testimony from 1986. Okay.
Yeah.
This is really an "I told you so" thing, and I find this really infuriating that there's even still denial going on. Come on, all right. This is important.
Well, there's a reason for the denial. It's political, right?
Absolutely.
Well, it's not just political; it's also economic.
Yeah,
I mean,
I think
this is one of those things. It was very appropriately called an inconvenient truth. It doesn't get much more inconvenient than saying that our entire economy is based on a system that's going to destroy the planet. That's a problem.
You think?
I think it starts out as the resistance starts out economical and becomes political.
It became ideological, yes,
Yeah.
but that's part of the whole.
The part of the part of the ideological split is do you care about how much you've got now, or do you care about everybody's welfare down the road? Okay,
right.
Greed, hatred, and delusion, the 3 poisons.
All right, Rich, what do you have for us?
On a lighter note, I just ran across this. This is a little one of these time-driven. Hang on.
Yeah.
One of these time-driven timeline animations with a bar graph that charts the evolution of web browsers, specifically desktop web browsers. And you can see it. I'd forgotten. It goes back to 1993. Dudes, before 1993, we couldn't do this, right? And I remember my son using, text, essentially text browsing. And I remember the first browsers, I wouldn't have been able to tell you what it was. It was Netscape.
Yeah, Netscape.
That's the first one I used. Okay. And this goes in the evolution. You can see Microsoft Internet Explorer dominating the landscape for quite a while with over 90% of the market share. Okay. And then Chrome creeps in and slowly but surely takes over the whole
Well, and
thing.
Firefox is taking, and they do along the timeline, they show Microsoft getting the antitrust flack,
Yes.
and their share is coming down, and
Right.
Firefox is
Yeah.
creeping up, and
Yeah.
then Chrome slips in.
I saw a couple of different versions of this, and I like this one, not only because it comes all the way out to 2022, but because of that annotation. Where, first ones I saw didn't have any annotation, and I was wondering, and I was vaguely remembering that there were lawsuits over business practices from Microsoft with bundling its browser, and the annotation includes all that.
Netscape
amusing.
Navigator. That's what it was
Yeah.
called. Remember?
With
Yeah.
a pulsating end in the corner.
Yeah.
I think, I did Netscape, and then I was Firefox / Mozilla is Firefox. Is that
Yes.
right?
Yes.
Yes.
I was Firefox for a while, and then Chrome came along, and it's in the family. So I did that. I have, for one reason or another, have resisted Internet Explorer from day one.
Well, now they have
I
a
hated
different. They
Explorer
have a
the
different
whole time.
browser, don't they? They have
Yeah, because if everything else fails, rename it.
Yeah.
That's called Edge.
Edge. I like that. So you're using Chrome, Rich. What do you use, Kathy?
Yeah.
Safari?
Mostly Safari. Yeah.
Yeah, me too. What about you, Alan?
Firefox.
And-
Been using Firefox since pretty much it became available.
And you're on
that's
a
my browser.
Mac, right, Alan?
No, I'm on Linux.
He's on Linux.
I was
Okay, of
I was on
course.
Mac for years, and I'm on Linux now, and I'm using Chromium for this Riverside app that we're using to record the episode because that's required. But
And what about you, Brianne?
Safari.
Yeah. All right. So now here's another question.
Wait a minute. How about you, Vincent?
He
So
said
I have the
Safari.
show notes in Safari, but the Riverside's running on Chrome, and I do. I use both. Chrome I use for different things, and I think I'm mostly Safari, but for Chrome I still use a lot. And of course, you always get a pop-up saying, this could be better in Chrome. It's okay. I don't want to change over.
as the notes on this video point out, this video doesn't include mobile browsers. If it
Yes.
did, then Safari would probably take over the whole thing after about 2008, because now most people browsing the web are on mobile.
But
And if you poke around, there's another version that does contain phone browsers.
But isn't Android the dominant platform? So they use.
I guess, yeah. So they use Chrome or some variety.
Yeah,
of
They
they
use
Chrome.
probably use Chrome. So I would say Chrome on mobile is the most.
But Safari is a big share of it. So it would be like Chrome and Safari
Yeah.
would be your mobile browsers.
All right, here's what I want to know, Alan. How many tabs do you have open on your browser?
Right now, I have 4.
No, not bad. Brianne, what about you?
I'm very lean about tabs.
At the moment, I just have the show notes and Riverside.
Very good. Rich.
At the moment, I just have 3, but I have to elaborate. I've got the show notes, and I've got Riverside, and I also have, because I just never close it, I've got the Google Drive Home tab open, but I also separate the tabs. So I've got 2 different windows. I've
you
got
got
Riverside
windows,
in
yeah.
one window, and I got the show notes in another.
Oh, and I should point out, Riverside is running in a Chromium window of its own, and
Yeah.
then I have 4 tabs in Firefox for everything else.
How about you, Kathy?
So I have 5 tabs in Safari and Riverside in Chrome.
Wow. So you win.
I had all the papers open, and then when I couldn't find my pick, I closed a bunch of them.
Yeah, if you had asked me at, say, I don't know, 2:15, 2:30,
Yeah.
that would not be the answer that you got.
Oh, yeah.
More
And here's
tabs
another
open.
paper I can.
I had a ton of tabs open
Yeah,
earlier.
so that's mostly when I see people, I look at their browser, they have 20 or 30 tabs open. And sometimes they never close most of them, right? They just leave them up there because it's convenient to go back. So
how many tabs do you have open on your phone? I have almost 100 open at the moment.
Oh, wow.
Yeah.
Yeah.
They never close them, right? Because when you go to a new
Right.
page, it leaves the old ones open. Yeah.
Right.
I'm always amazed when I go back and look at it. Whoa. I haven't looked at that
Yeah,
in a
it's
long
kind of
time.
fun. You go back in history and relearn stuff that
It's
you
a
looked
way
at
back
before.
machine.
Yeah.
Yes, the modern browser usage. Right. Well, I have Safari has one tab open now with the show notes, nothing else. I mean, during the course of the episode, there were more tabs with some of the papers and stories, but it does use resources to have many tabs open. And even though modern browsers are good at managing them, when I always say to people, when you're recording, we want to give as much resource as possible to the recording program. So, but many of our guests don't do that, and they have email popping up and ding, 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 ding every 2 seconds, And Dixon also probably has a billion things, and he's not here, so he can't defend himself. All right, we still are not finished with picks. I'm sorry. Alan, what do you have for us?
I have a short interactive story. It is technically a video game because it runs on your computer, but it runs actually in your web browser. And you can play this in about 5 minutes. It's an interactive work of fiction. The game aspect of it is that your task is to turn off a robot. So it is possible to fail at it. But it's fairly straightforward. And I made this for something called a game jam, which is a challenge to produce a game in a particular period of time with a set of constraints. And the constraints of this one were quite manageable. It had to be of a particular format, and
It's funny.
that's spelled out on the page. So
I like the title.
and that has a NASA theme. It has a NASA theme to it. So go ahead
Alan,
and enjoy.
was this a competition?
It nominally, sort of, kind of. They did. Some of these actually do have. There's a big one going on right now that's or starting. I think it's starting in September. There's one that's make a Halloween game, and it runs all of the month of September, and then in October they judging, and they do like thousands of dollars worth of prizes for the thing. But this was just for the glory of doing it and seeing what other participants.
Well, I love it, Alan. I've played it 3 times. Okay, the first time I played it, I kind of didn't know what was going on, and so my answers were kind of sort of more or less random. Though I think I kind of was catching on towards the end. Second time I played it, I decided, okay, I'm going to be a compassionate human being, and I'm going to treat this robot with respect. Okay, and I succeeded. And the third time I decided, okay, I'm going to be a jerk. And I failed. It's outstanding. I love it. And the questions and the responses are really great. It's very nice. I really like it.
So you got both endings.
Cool.
Good.
That sounds so
I'm looking
fun.
forward to
I'm
it.
definitely
Yeah.
doing that later.
So it's called I Am Promise, by the way, in case I didn't give the title.
Oh, by the way, Brianne, your Wordle, was it Wordle? No, it was something else.
Oh, is it Redactyl?
Redactyl is people are writing saying I'm addicted to Redactyl because of Brianne.
I got addicted to Redactyl very quickly after being introduced to it.
I do, like Alan, I picked something I made. This is a video called Polio in New York, which I did 2 weeks ago, I think. Yeah, maybe. Is it 2 weeks? It might have been last week. I don't remember. But it's just an explainer, a 20-minute explainer of the case in Rockland County, a little bit of history about why it's happening and so forth. So if you want to explain it to someone, you should check that out. I think most listeners have already watched it, but if not, check it out.
Vincent, what do you use to make this? I like the way that you alternate slides with you rather than having a thumbnail of you up in the corner. How did you do this?
Well, I have this fancy studio here in Manhattan. So I have a camera in front of me, and I have a
you could just
laptop.
edit this.
So the camera and the laptop go into a recorder, and so I'm recording both the video of me talking, and then the it's like giving a PowerPoint or something, right? And that's recorded separately, and then I edit it. Yeah, I put it on a timeline in a video editor, and I cut back and forth to them. It's very so I have everything I need here, and I know how to do it really quickly. So I actually knocked that off in 2 hours on the Friday morning and posted. I had put the slides together on the train the night before, so I'm really learning how to take advantage of train commuting. It's much easier to make a lecture on the train than it used to be in my car.
Mm-hmm. Yeah. Do you use ScreenFlow to edit them together?
I don't. I use ScreenFlow when I'm in class, and I use it to capture the slides and audio as well.
Right.
But this I used. I have a video switcher here that has a built-in recorder. It has eight inputs, and they're all recorded separately. So then, I have a little drive that I can take and get the videos off of that as well. But you could use ScreenFlow to do the laptop recording separately and then combine it with the video. Or, so the other thing you could do, because I have a video switcher here, I could actually manually switch when I wanted to go to the slides. But then you would see me reaching for the switcher slightly, and I don't really like that each time. And then sometimes I don't switch it at the right point, and this way I can, control it. So video editing is not hard.
I think you could probably do this. Without the extra hardware.
Yeah,
in
you
OBS.
could do OBS too. Yeah, you could use if you put your how are you going to get? So I have a camera. Yeah. Yes, you two different captures. You capture your video camera and you capture the screen in OBS. You could do that as well. Yeah, I don't use OBS, but I have other solutions. So, and that's what I've done to make a number of videos. And the whole point of me being here is to make more of these. So I thought I should do it. But it's also something I've thought about for 40 plus years. So
it turned out very well.
Thank you. All right, that's it for TWiV 925. Show notes are at microbe.tv/TWiV. You can send your questions, comments, your picks of the week to TWiV@microbe.tv. And if you enjoy our work, you could support us financially, microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Alan Dove is at alandove.com, Alan Dove on Twitter. Thanks, Alan.
Thank you. It's always a pleasure.
And Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. It's great to have everybody together. This is good.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.